All right, let's get into it. Today, we're jumping into a high-stakes investigation. Think of this as your ultimate crash course, your exam review for cracking one of the toughest cases in microbiology, identifying the tiny culprits behind a disease. So here's the scenario. You're the clinical detective. A sample lands in your lab, and a patient's health depends on you. Your mission? To grow, isolate, and name that pathogen. The clock is absolutely ticking. This isn't just about memorizing facts. It's about seeing the clues and knowing what they mean. To solve this, we're going to follow a 5-step game plan. We'll start with the initial puzzle, then set up the right environment for our suspect to grow, pick our tools, read the visual evidence, and finally, look at some of the really tricky cases. Let's get started. Okay, first things first, and this is the most crucial point. You can't identify a suspect you can't see, so your number one job is to take this invisible microbe from a patient sample and culture it. You gotta grow it until it forms a visible colony that you can actually work with.
So how do we do that? Well, we have to build the perfect environment to coax our little microbial suspect out of hiding. Just like any living thing, these bacteria have their preferences. They need the right temperature, the right atmosphere. If you get this part wrong, your suspect might never even show up.
Now, this is a great way to think about it. Bacteria are kind of sorted by their favorite temperature. You've got the psychrophiles who love the cold and the thermophiles who love the heat. They're fascinating for sure, but they rarely cause problems in humans. For us, in a clinical lab, we are laser focused on that middle group, the mesophiles. Why? Because they thrive at moderate temps, which, you guessed it, includes our own body temperature. And that brings us to the single most important number you need to burn into your brain: 37 degrees Celsius. That's human body temperature. It's the cozy, perfect environment that most of the bacteria that make us sick are totally adapted to. In the lab, this is your default. This is where you start.
But of course, every great detective knows the exceptions to the rule. And for your exams, these are pure gold. For example, Campylobacter actually likes it hotter, around 42 degrees. And this is a key trick. By incubating a stool sample at this higher temp, you help Campylobacter grow while you knock back a lot of the other gut bacteria. Then you have guys like Yersinia and Listeria, who prefer things a bit cooler, more like room temp. And for Listeria specifically, remember 25 degrees? That's the magic number where you'll see its signature tumbling motility, a dead giveaway.
All right, we've set the thermostat. Now it's time to open up our toolkit. And in microbiology, our most essential tools are the culture media, or agars. Just think of them as different types of bait that we use to lure out different kinds of microbial suspects. Every investigation starts with a standard set of tools. Blood agar, that's your workhorse. Almost everything will grow on it. Then you have chocolate agar. And no, you can't eat it. It's basically cooked blood, which releases extra nutrients for the really picky, or fastidious, organisms like Neisseria that just won't grow otherwise. And then you've got agars like CNA or PEA. These are your specialists, designed to only grow gram-positive bacteria while stopping everything else in its tracks. Now, this concept right here is absolutely critical. You have to get this. Selective media, it acts like a bouncer at a club. It has inhibitors that only let certain types of microbes through the door. Differential media, on the other hand, is more like an indicator. It lets lots of things grow, but it makes them look different based on what they're doing chemically. And here's the kicker. Many of our most powerful agars are actually both. And this is where the real detective work kicks in. We've baited our traps with selective and differential media. Now, we've got to interpret the visual clues they're giving us. The color, the shape, the appearance of the colonies, that's our evidence. Okay, let's start with a true classic, McConkey agar. It's designed to grow gram-negatives and show you if they can ferment lactose. The rule is super simple. If you see bright pink colonies, bam, you know instantly that your suspect is a gram-negative that ferments lactose. If the colonies are clear or colorless, it can't. It's a powerful first clue. Next up, hectoin agar. The big thing we're looking for here is H2S production. If your organism produces hydrogen sulfide, it reacts with an ingredient in the agar and creates these really striking black colonies. This is basically a smoking gun for pathogens like Salmonella. Seeing black on an HE plate, that's a huge break in the case. So we've covered the basics, but let's be real. Sometimes our suspects are masters of disguise, or they need some seriously special conditions to grow. This is where we call in the specialist toolkit, and we dive into the unique world of mycology, the study of fungi.
So what happens when your standard plates come up empty, but the patient's symptoms are screaming a specific diagnosis? Well, that's when you reach for these special tools. If you suspect Neisseria meningitis, you grab a Thayer-Martin plate. For weird pneumonia, you might use BCY agar to hunt for the elusive Legionella. Each one of these is a highly specialized trap designed to catch a very specific bad guy.
Now, let's switch gears to fungi. The secret to finding them is to create an environment that they love, but bacteria absolutely hate. And that's exactly what Saborod's agar does. It has an acidic pH of 5.6. Most bacteria can't stand this acidity, which gives the slower-growing fungi a chance to finally show up and be seen.
And when you're talking about fungi, one of the most important and definitely most testable concepts you'll ever learn is dimorphic fungi. These guys are the shapeshifters of the microbial world. They can be a mold or they can be a yeast, and the trigger that makes them change is just temperature. And this shows that switch perfectly. At cooler, room temperatures, they grow as a fuzzy mold. But if you take that same culture and incubate it at body temperature, 37 degrees, it transforms into its yeast form. There's a classic mnemonic you have to remember: mold in the cold, yeast in the heat. Proving they can do the switch is how you confirm their identity.
All right, let's do a quick-fire recap of the biggest clues. 37 degrees is your default incubation temp. On Maconchia agar, pink means it's a lactose fermenter. On HE or SS agar, black colonies scream H2S producer, like Salmonella. Dimorphic fungi, they're mold in the cold, yeast in the heat. And if you're hunting for any fungus, you start with Saborod's acidic agar. These are the core facts you just have to know. So there you have it, the foundational toolkit of a clinical microbiologist. Now you know how to set the scene, how to choose the right tools, and how to read those critical first clues. So the next time you're faced with an unknown pathogen, which piece of evidence, which color, which temperature are you going to use to crack the case wide open?